We're really proud to welcome a fantastic speaker for our 6th annual lecture. It's Dame Sue Hill this evening. It's a real privilege to have her here. Dame Sue started her career as a healthcare scientist in the NHS, specialising in respiratory medicine. She got her PhD in Birmingham and worked for many years as a scientist within the NHS and the University of Birmingham with a stellar academic career in respiratory medicine. She then moved somewhat out of the academic sphere into the strategy sphere and became the first Chief Scientific Officer of the UK in 2002, transferring across to NHS England in 2013 when it reorganised. Sue's been instrumental in many programmes over those many years, but has really been absolutely essential in establishing Genomics England and has been a really driving force in setting up the NHS Genomic Medicine facility. And she tells me she's been travelling the world exploring other systems and reassures me that we're still a little bit ahead of the game in this sphere. So it's a huge privilege and pleasure to welcome her here today. Sue was awarded a fellowship of the Royal College of Physicians and was made Dame Commander in 2018, and it's an enormous pleasure to welcome her to Oxford to be our 6th annual lecturer. Dame Sue, thank you.
Good evening. I'm absolutely delighted to be here, and thank you so much to the Centre for Personalised Medicine for inviting me to give this annual lecture. It has indeed been quite an interesting few weeks for me. I've just come back from a 2-week immersion visit looking at genomics in Australia. And earlier this week, I gave a talk at the Academy of Medical Sciences in Norway about the Genomic Medicine Service that we've been introducing into the NHS to really, hopefully, drive the NHS, so real-world clinical evidence, informing research and development to drive improvements in patient care. And I hope what I'm going to tell you tonight will be that you'll understand the infrastructure we're putting in place, the opportunities that are there, but also the opportunities of thinking about how we might do something similar in other areas to benefit the patients and the public that indeed much of our research is aimed at. So I'm going to start by taking this quote from Eric Topol's review that he did for Secretary of State and published earlier this year, which really looked at the impact of emerging technologies on the healthcare workforce. And he's made this quote many different times, said, "Genomics is probably the biggest breakthrough in the last 50 years. And for many years, it's been talked about that medicine is going to be personalised, and we're finally starting to get there." But what he recognised is there's a real opportunity to deliver healthcare on a far more rational, efficient and tailored basis, but that we have a huge job to do to both educate our patients, our public and our healthcare professionals. Genomics is now a global initiative, and since 2013, the governments of at least 14 countries have invested over $4 billion US in establishing national genomic medicine initiatives. And you can see one of those initiatives here is in the UK, which I'm going to talk about in a moment. There are, if we look across the world, some 56 different genomic initiatives, more growing each day. Very few of those initiatives link what are genomically related research projects with evidence to inform healthcare delivery or indeed individual patient care. And I think that's part of our challenge. But what the paper that I was a co-author on earlier this year said very clearly is that sharing data, tools, experience and knowledge to create a global learning health system is essential if we're really going to get the maximum benefit from genomics for the people that we're going to serve. And I think that's one message that I'd like you to take home tonight. We need to think outside of the shores of the UK if we're really going to give our patients the best benefit, but also in terms of our research endeavours. I'm going to set the scene now for the health system before I go into more detail about what it is that we've been doing. So first of all, within the National Health Service, we have recognised the potential of genomics to be transformative, to be that disruptive innovation, to move us from both a more personalised based healthcare system that's focused on late disease diagnosis to one that's much more about population-based health and earlier intervention. But what genomics enables us to do is to move from protocol-based medicine to data-driven healthcare, from volume-based healthcare to value-driven healthcare, and to move away from a one-size-fits-all approach to treatment, where we know that between 30 and 50% of individuals who receive medicines don't actually get a benefit from them, and 1 in 15 people end up in hospital with an adverse drug reaction, to more better care, tailored and stratified treatments. And our challenge is how we match the genomic profile with the other diagnostic profiles, other clinical information, together with the range of treatments that might be available to treat an individual patient. Earlier this year, the NHS set out its long-term plan, and this was a vision for the next 10 years, but a strategy for spending the extra 20 billion that the government put into the health service to ensure that it was sustainable over the next 5 years of delivery. It focused on trying to address how there can be systemic improvement in a number of clinical priority areas for the healthcare system where outcomes were poor. Outcomes were poor for patients, and the cost of delivering the services in terms of individual hospitals and other providers in the healthcare system was rising. As part of those long-term plan discussions, I managed to secure a tripling of investment in genomics over the next 5 years. And what we've got agreement to do is to fund in a new central resource of money is to sequence 500,000 whole genomes of people who are being treated as part of routine care in the NHS and funded by the NHS. And through that to improve recruitment to clinical trials and to support ongoing research and development. Secondly, that all children with cancer would be offered whole genome sequencing, even though at the moment the whole genome sequencing may not be necessary for the treatments that are available. But what we want to do is make sure we've got the best possible genomic baseline to drive drug discovery so that we can start to turn the corner in terms of improving outcomes for people, for children who develop cancer. Two other areas are extending the access to molecular diagnostics for people with cancer, so to give people with cancer a more comprehensive assessment of their the genomic profile of their tumour. And at the moment, it's very ad hoc across the National Health Service. But if we get this right, we've got the opportunity to align it with clinical trials and to really drive pharmaceutical companies to come to this country and to invest and to align their clinical trials with our National Health Service. And then we will also expand access to genomic testing for people who have high circulating fats in their blood associated with a condition called familial hypercholesterolemia, where still a significant number of people die from cardiac death because of undiagnosed familial hypercholesterolemia every year. And at the bottom, it recognises that we need to link and correlate genomic clinical data and patient data. So this is the NHS focus, but there's a much broader alignment across the UK government, although, of course, we're without a government at the moment. And this slide really represents the commitments that are already in train rather than new commitments that might be made by the incoming government. What Sally Davis's report, so the previous Chief Medical Officer's report on Generation Genome, really set out some of the gaps and some of the issues we needed to address genomic services and their access. And the government last year set out that in total it would fund 1 million genome sequences over the next 5 years. That 500,000 funded by the National Health Service and another 500,000 through the UK Biobank. So 1 million whole genome sequences. Overall, worldwide, there's projected to be 60 million whole genome sequences that will be performed by 2025. But also linked to this, and very much the brainchild of Sir John Bell, has been not only the investment in artificial intelligence, in digital data and other forms of bringing data together for patient and research endeavour, has been the establishment of the Accelerating Disease Detection Cohort. And as part of that work, individuals from the population will have a polygenic risk score developed. But you can see at the core, really, of the life sciences strategy that John Bell's been so instrumental on, has been this relationship between aligning real-world evidence with research endeavour, with industry for discovery and patient benefit. So what's the journey that we've been on? Well, this just builds on the history of genomics in the NHS. First of all, we've had genomics in the NHS since it was formed in 1948. It's evolved organically. There's been some parameters put around it depending on the government at the time, and also the focus on genomics for improving both our research standing in this country, but also improvements in patients. But where we were in 2012 was that we had 23 genetic labs mainly focused on rare disease, 35 molecular pathology labs mainly focused on cancer genomics, and 17 clinical genetics services. No national coordination or measures of activity or outcomes. They've evolved organically, no systematic approach to evidence or technology adoption, so it depended where you lived in the country, what you'd have access to, and often that related to the interests of the people employed in those services. We had escalating costs of single tests and an ongoing variation, inequality and inequity. And they weren't embedded in end-to-end patient pathways. Now, at the same time as that was recognised as something we needed to address, the 100,000 Genomes Project was announced by the Prime Minister of the time, David Cameron, as a project to really continue to put the UK on the world stage, both from its original discovery of DNA, but also from all the other endeavour that had been happening over many decades, with the plan to fully sequence 100,000 genomes from people and their families with cancer and rare diseases, so focused on those with the most unmet need, and to really help to understand in the clinical service what it is this would bring in terms of a greater understanding of a patient's condition and treatment needs. And also to help in a discovery approach to look and drive new drugs, new diagnostics and new scientific breakthroughs, and particularly gene discovery. This became a partnership between NHS England by Genomics England that was set up as a limited company with a single stakeholder, a shareholder, Secretary of State, with a coordination focus, and then linked to Public Health England and Health Education England. The NHS contribution to the project and the project as a whole in terms of its architecture is outlined here. On the left-hand side, in NHS England, we established and funded through an open procurement process 13 NHS Genomic Medicine Centres. Those centres worked across geographical populations of between 3 and 5 million, and with multiple hospitals within that geographical location, to both consent patients, including for the use of their data for multiple purposes, for collection of samples, clinical data, and through the consent process, helping patients understand the importance of their longitudinal health record being brought together with their genomic data to really increase the likelihood of getting a diagnosis. And then after sequencing of the whole genome, validation and reporting for clinical care purposes, and then the return of results to our participants. In the middle is the role that Genomes England played, which is they established a biorepository, a sequencing centre that was provided by Illumina in Cambridge, and then they developed an informatics architecture that created a reading library, not a lending library, with access to data through a secure firewall. And then to that informatics infrastructure, the sequence, after going through a bioinformatic pipeline, could be fed back into the NHS for validation and reporting, or could be accessed once access rights were granted to people in Genomes England clinical interpretation partnerships, so in research partnerships for research and discovery, or through a discovery forum with industry users. What we did on the right-hand side is we took a strategic approach to delivery that looked at the evidence of the impact of whole genome sequencing in the NHS, that we were reflective of the population that we serve. We created those strong partnerships and clinical leadership to mainstream genomics for the future, and we directed investment to support transformation rather than highly specialised centres, and we wanted to broaden the knowledge and use of genomics and develop the informatics infrastructure. And it was aligned with the NHS transformation model. In less than 4 years, the NHS Genomic Medicine Centres started to transfer service delivery. And I'm not going to go through all of this, but they literally brought together thousands of clinicians and clinical teams to deliver the contribution. They raised the awareness of genomics with over 50,000 people, including members of the population. They established genomic multidisciplinary teams to review those whole genome sequence results, to understand what the plan would be if there were variants of unknown significance, to establish whether they weren't pathogenic, and a mechanism to feedback. And this was across rare disease and cancer, established in a systematic way. On the right-hand side, what it did do is it really showed how this integral alignment of research and clinical care for patient benefit could be established, and how we could take a whole NHS approach to embedding the mainstreaming of care. In the bottom, though, we had to establish many new clinical pathways and models of care to deliver the contribution that are going to be essential for the future for embedding genomic medicine. And this includes fresh frozen pathways for cancer tissue acquisition. And this is because if we look at these series plots here, what we learned through the process is the usual fixation process in the NHS for cancer tissue is to drop it into formalin and fix it in paraffin, and then cut off very fine sections of it. If we do that, this is the outcome for whole genome sequencing. When you look at the data in more detail, you get a loss of some of the critical letters of the genetic code. On the left-hand side, this is what happens if the sample is just treated in a fresh frozen way, so not put into the long established pathway of formalin and then paraffin embedding. This meant we had to create new pathways right from surgical retrieval through to processing and extraction of DNA from fresh tissue. And in the middle, we had to look at different approaches to how we could move tissue, which was fresh and frozen, across significant geographies. And that might be from Cornwall up to, for example, Bristol or beyond, so significant areas. Significant re-engineering. And what I can tell, and I normally tell my own personal story here, is when I took over as responsible for the NHS contribution to the 100,000 Genomes Project, I didn't expect to be a participant myself. And I'm a participant in the cancer arm. I had breast cancer diagnosed in 2017, surgery in 2018. And because my whole genome was sequenced, or I asked for it to be sequenced and to be part of the study, my surgeon kept me on the table for an extra 45 minutes to make sure the tumour that they'd resected was not put in formalin and somebody actually came to collect it. So there's a lot of work that needs to happen to really understand how we re-engineer. What I can say, it didn't affect my treatment, but it's certainly on file if I develop any other symptoms for the future. So where are we going? So what did the project show us? We had 1,200 disorders with unmet clinical need in rare disease, over 30,000 families, most of those with intellectual disability or neurological conditions, but a significant number of those with cardiovascular disease and, for example, with hearing. What we knew is that it ended a diagnostic odyssey for some, and on average 1 in 4 patients got a diagnosis that they wouldn't have got on the National Health Service today. In some areas, in ophthalmology for example, this was as high as 1 in 2. What we know though from this data in the middle here, the more we're able to characterise patients using a human phenotype ontology system, the better the diagnostic yield, because the greater the genotype and phenotype correlation. In cancer, there were 24 cancer types included, 18,500 participants. And what was found in cancer was 50% have a known actionable or potentially actionable gene that may enable them to be eligible for clinical trials. So it demonstrated the value, again, over and above standard of care. What this data is doing now, because it sits in Genomes England de-identified research database that they created, there's over 100,000 genomes with associated secondary data that includes hospital episodes, patient-reported outcome measures and diagnostics, and a significant amount of clinically interpreted data that's come back from the NHS in terms of the variants that have been reported and the new treatments that have been and the treatments that patients have received. In the bottom, Genomics England have also established a discovery forum with now over 100 companies involved. Why this is really important is on the right-hand side of this slide, because what the involvement of both researchers and companies enabled was diagnostic discovery. So over 100 participants got a diagnosis because of those researchers and industry scientists looking at their whole genome sequence, and particularly because of the variants of unknown significance that were generated. And the bottom is just an example in kidney disease that I'm not going to talk through. But on the left-hand side is a story about Jessica, who'd had, in her 4 years of life, had over 135 different episodes associated with a multiplicity of hospitals at a vast cost to the system. She took part in the project, and she was a gene was identified that interfered with the glucose transport into the brain. Essentially, to control her seizures, All she needed was a fairly simplistic change in diet, so a ketogenic diet, and her seizures have reduced considerably. The reason I put this in was because one of the reasons I was able to make the argument for the investment in whole genome sequencing is it's not all about expensive drugs at the end of the day. It can be a rather more simplistic intervention. There's more to do though. We've got to complete the validation and reporting of primary findings, and particularly when we reanalyse and the balancing the need for the demand for new people in the system versus those that are existing with a whole genome sequence and needing to reanalyse when there's new gene discovery. When do we do that? The second is feeding back additional findings and also pharmacogenomic profiles, and we need to know the impact on NHS services. What criteria should be introduced for incidental and additional look-for findings, and how does the National Screening Committee play a role? And then understanding, because we can do this from the use of the whole genome sequencing, what polygenic risk scores might mean in terms of the impact on services and the requirements of individual patients. So what we've done then is create the infrastructure for precision medicine in the NHS Genomic Medicine Service. We launched in 2018 the NHS National Genomic Medicine Service with an aim to serve the 55 million people in the population that the NHS in England serves. National approaches to how patients are consented, to how patients and the public are involved, common standards, specifications, protocols, contracts, activity monitoring and pricing frameworks. And for the first time, we'll have money in one place to support cancer genomic analysis. And consolidated National Genomic Laboratory Network made up of 7 genomic laboratory hubs. If you remember, I said there were 23 for rare disease and over 35 for cancer doing some sort of testing. We've now got 7 hubs, and they are charged with also driving rapid adoption of technology, with partnerships across local providers to ensure that we've got end-to-end, a single national testing directory that's mandated for use in the NHS, covering all technologies from single gene to whole genome sequencing. And then that continued, if patients consent, that continued movement of de-identified data into the knowledge database that will be curated and annotated by Genomes England. This is what it looks like. Critical to this at the very top are NHS Genomic Medicine Service Alliances, the alignment with all other clinical services that are critical to deliver genomic medicine. And then on the bottom left, having informed patients and clinical teams, especially around the new patient consent model, where every patient in the NHS receiving a genomic test will be offered the opportunity to participate in research and their de-identified data to enter into the database. 7 genomic laboratory hubs, that genomic test directory that I spoke about, and then whole genome sequence provision really procured in a partnership with Genomes England, who will continue to provide a bioinformatic pipeline. And when patients have consented, data will deposit into the UK Genomic Knowledge Database to support ongoing research and discovery. There's a focus as we move forward on embedding genomics in end-to-end patient pathways, from prevention through to treatment selection and to monitoring reoccurrence, and to providing integrated diagnostic reporting to inform personalised care. We're evolving the role of the participant and patients within the system. They've been very critical in terms of providing input into the access of researchers, of industry, indeed sometimes clinicians, to that data that's been curated by Genomes England. And within the NHS, there's been genomically facing patient and public engagement groups, which will continue as we move into the future. We're evolving, as I said, the whole infrastructure for the future, and a critical part with it will be at what was at the top of my slide, evolving the infrastructure for the future with 7 NHS Genomic Medicine Alliances, covering the geographies that have been established for the 7 genomic laboratory hubs. And you can see the one that relates to this area is this one in here. And this is just a working title for what this alliance might be. So the Genomic Medicine Service Central Alliance. The responsibilities of the alliance are inclusive of clear governance structures built on collaborations across provider organisations, all provider organisations in those geographies, from primary through to secondary and tertiary care, patient engagement and involvement, coordinating implementation of genomic medicine and ensuring equitable access to the testing within the test directory, and creating strong clinical leadership and new clinical roles to drive new models of care across the geography, and to really understand the workforce development that's required and how MDTs can be supported, but establishing research networks that I'll come back to, and coordination of input into future genomics projects and broader NHS long-term plan. So we will be funding research coordination within the new Genomic Medicine Service Alliances. And importantly, they will be charged with how we drive personalisation of care and medicines optimisation, particularly because this is a way in which we can get real savings into the future, and with a drugs bill that's due to increase. So this is the GLH in practice for this area, the Genomic Laboratory Hub. It covers 19 million. The governance is provided by Birmingham Women's and Children's. There's an integrated governance board. And within this Genomic Laboratory Hub geography, the geography will be delivering many of the specialist testing in rare disease across cardiology, endocrinology, ophthalmology, haematology, inherited cancer, metabolic, mitochondrial, MSK, NIPD, and respiratory. So one of the biggest groups in the country, and there will be consolidated cancer panel delivery. So the standardised approach, and this is where you can get the information, sets out in a test directory the what, the where, the why, the when and the how of each genomic tests. It covers a range of different clinical indications which will drive ensuring equity of access. The standard testing is being standardised across the country in terms of the technology and the target genes and its relationship to the clinical indication. And then it's linked to activity and outcomes on testing for the very first time. And this test directory will be updated annually on the basis of the review of the emerging evidence, which is why it's so important to align research and development alongside the clinical system. We're standardising the panels that are used in rare disease by using a consensus approach and a tool that's been developed by Genomes England called Panel App. And you can see that across the country here, all of our genomic laboratory hubs do core tests, but then depending on what type of very specialist rare disease test it is, there may be only 2 providers or 7, or in the case of neurology, or 7 because of the volume. But this was a very helpful way of getting to something that enabled us to understand what the high level of evidence was for including particular genes on a gene panel that will also be applied to whole genome sequencing as we go forward. We're currently standardising cancer panel testing, and this is really exciting. We've been in the NHS focused on single gene tests, then moving to small panels, but now we're moving to extensive next generation sequencing panels with both DNA and RNA based targets. We've looked at future-proofing this for the next 2 years, including for targets not only for treatments we've got now, but for clinical trials that are in the pipeline 2 years ahead. And we've been working with CRUK and other agents to ensure that the testing in the NHS will align with the trial requirements, so there's a speedy access to clinical trials. And you can see we're particularly focused at the moment on how we introduce testing for the which will underpin the new tumour agnostic drugs that will come into licensing into the NHS early next year, called NTRK gene fusions. But these are all the biomarkers that we're also building into our approach within the NHS. If a patient consents, then this de-identified data can be available within the research database. We've just established a rapid exome service for NICU, PICU before we move to whole genome sequencing. A nationally commissioned one service provided in the southwest for acutely unwell babies. And the genetic diagnosis found since the service was launched on the 1st of October show that a genetic diagnosis has been found in 36 cases at the moment, but that's likely, in terms of all the other work that's ongoing, to increase to over 40%, and the results have been issued in a mean of 9 days. This is for a national service. And the outcomes have meant referral to specialist service, annual screening for diabetes, or, for example, redirection to palliative care. I mentioned that we've got funding in the NHS to introduce whole genome sequencing into routine clinical services. We'll start with 21 conditions that are in here, and that includes, for example, cystic renal disease and some other child onset diseases. But we'll be rapidly moving next year in 2020 to another 25 that includes adult onset neurodegenerative disease, some mitochondrial disease, and, for example, thoracic aorta aneurysm or dissection. On the left-hand side, we're going through a systematic approach to going live, and this service will be live in the National Health Service in the next financial year, so starting in April 2020. These are the cancer conditions that are going to whole genome sequencing, so sarcoma, acute myeloid leukemia, and all paediatric tumours, as I said earlier. Our focus is maximising the impact of mainstreaming genomics as we go forward, recognising that there still will be a continuum of testing from single gene through to whole genome sequencing, that we need to focus on primary diagnosis, but recognise at the right-hand side here that we have an opportunity to really drive secondary diagnoses and population interventions, and some greater feedback around additional findings on pharmacogenomic profiles or polygenic risk scores. What we will be doing in the strategy, though, is recognising, and this is some work that was being generated by the technology developed here in Oxford, at Oxford Nanopore, that actually shows that as we move forward, to get a diagnosis, an individual patient, we may need to start to use, for example, for whole genome sequencing, both long read and short read technology, if we want to really enhance the diagnostic yield of the individual patient. There's some real opportunity with the Oxford Nanopore devices as we move forward to the delivery of more point-of-care genomic testing. So this is all supported by a new national genomics informatics system that enables that population right at the end here of the de-identified research environment, but also in the middle here of a bioinformatic pipeline that includes a variant store that will continue to be populated both from non-whole genome sequencing and whole genome sequencing, which will enable, for example, the submission of variants with genotype-phenotype correlations to organisations such as ClinVar. And at the right-hand side here, our challenge is how we link up the whole of the NHS with this system that supports ordering. What's really exciting is that we have got agreement to introduce a research framework that will be called the NHS Genomic Medicine Service Research Collaborative, where work will happen between NHS England, Genomics England, NIHR, industry, academics, the NHS and other partners to support and enable increasing volumes of high quality genomic research in the NHS, and with access, for example, to samples, to data and to recontacting patients. A critical part of this will be the steering committee in the middle here, which will include representatives of each of those genomic medicine service alliances, and they will be expected to create 7 strategic service research networks across their geography. And then there will be a research network advisory forum to try and align all of the research alongside this. What this collaborative will be driven by is an ongoing commitment to align and deliver the strategic priorities of the NHS and of the UK life sciences sector, but with research and industry collaboration really driving the benefits for patients, so with all outcomes fed back to the NHS to support improvement of care. All genomic data will be deposited into the genomic research environment that I've talked about, and will collate a register of genomic research activities that are being supported by the NHS, where data, samples and patients are going to be brought together to support research projects that could be across the whole of the country rather than necessarily in an individual system. We're obviously working with all the elements to ensure that this could be aligned with existing processes and addressing the intellectual property issues. So this will create the platform for precision health. This is all about integrating genomics and diagnostics to drive personalisation and the 4 Ps of personalisation at the right-hand side here. So better prediction and prevention, a more precise diagnosis, more targeted and personal interventions and a more participatory role for patients. What we recognised around the outside as a number of things that we need to do. What we can do and what we can learn is if we analyse the whole genome, we can generate additional findings. And these are the findings that will be fed back here in the 100,000 Genomes Project, a rather conservative list compared to those in the US, for example, around tumour dispositions, syndrome genes, also familial hypercholesteremia. We will look at how that may be expanded so that this really supports the more predictive and preventative healthcare agenda given the investment. The potential for personalisation is huge, though, in terms of us understanding the outcomes and response from treatments, to really understand those people that will have no benefit and possibly toxicity, those with benefits and toxicity, those with benefits but no toxicity, and no benefit and no toxicity. This is really important if we want to personalise medicine, and we have to do this in a systematic way and introduce this across the whole of the NHS so that we deliver tailored treatment and improved outcomes. What we're trying to do is to create a framework to match genomic variants with the intervention. So whether that's existing medicines, whether it's repurposed existing medicines, new drug discovery and rediscovering previous candidates, our approach to targeted medicines, including advanced medical therapeutic products, how we avoid adverse drug reactions, and what other interventions. So we're looking at all of this to create a framework for the National Health Service, recognising if we become increasingly precision-based, this will be more focused on carefully identified subgroups within the population through their genomic profiling. We're already doing this for AML, for example, to ensure that the testing aligns with the NICE approved AML therapies. We're proactively planning for the new introduction of therapies, and that is work with NICE at this end of this process to really understand how we can describe what will be more cutting-edge technologies in the context of the value chain for new medicines coming into the system. How we can also align our genomics programme with both the Access to the Cancer Drugs Fund and the Managed Access Agreement for other drugs, so that we can drive from genomic variant into medicine, into routine commissioning very quickly. And this will be by aligning real-world information. We're already working, as I said, on tumour-agnostic drugs, the new NTRK candidate drugs that target tumours with this variation. And this is working to bring together the whole community to make sure that we're able to systematically introduce this during 2020 to agreed target groups. What I do want to mention, in the advanced medical therapeutic world, we have therapies like CAR-T therapy. It costs the NHS a lot of money, in excess of 250,000 per individual patients. Some patients do very poorly. Many of these patients have leukemias or B-cell lymphomas, for example, other diseases. And what we're starting to consider is how we might actually fund whole genome sequencing in those patients to help us better understand why they may or may not respond to treatments, because the cost of the testing is relatively small compared to the treatment. And that isn't the approach that we would usually take, but it's starting to drive a personalised medicine approach. What we know is if we get this right, patients can really benefit. And this is a child with a brain cancer where the urgent review of their whole genome sequence revealed a novel BRAF fusion that meant that the clinician could apply for some particular type of therapy following discussion with experts in other countries. And this illustrates the need to work beyond these shores. And the patient actually improved after they'd had this therapy compared to conventional therapy. What we've got to do is systematise this and introduce standardised end-to-end patient pathways. And that's what we're working on. And that's what those Genomic Medicine Alliances will do, also supporting rapid adoption. Whether that's in breast cancer, in neonatal onset diabetes, where we don't do well for the whole population, or whether it's thinking about a surgical conserving set of techniques in colorectal cancer. Interestingly, down here, in the analysis of the 100,000 genomes participants so far for pharmacogenomics, 83% of those genomes that were looked at had actionable information about drugs that patients shouldn't receive. Think about what that means in terms of ensuring that we've got a personalised medicine approach to stop people getting either adverse reactions or appropriate readjustment of dosing. We are working on a 43 drug gene pair panel to introduce into the NHS, focusing on drugs that cost the NHS a lot of money, but that will enable us to reduce the toxicity in the areas where we know there's significant evidence to support their introduction, to reduce toxicity or to readjust dosing. And we expect to pilot that in 2020. So as I move to the end, we need to think about a multi-level approach to personalisation in in the conditions. This is just one example taken from respiratory care, to think about systems networks, whether that's at the bottom on this slide in terms of the genetic network, the biological network, the clinical network, or the environmental network. We have to think about and bring this all together if we're really going to influence an outcome for a patient. And this is just one example from the west coast of the States, where they've started to look at how they could introduce a near future personalisation pathway for cardiovascular care. From the blood that's analysed to determine CVD events, to biomarker monitoring, to genomic and biomarker data to select the most effective medicines, to drug platforms to enable designer drugs for personalised treatment or the matching with drugs that are available, and then different devices to measure whether a patient is taking treatment. This is what we'll need to introduce if we really want to move into personalised medicine at scale and at pace across the NHS. And we'll need to look at this in individual conditions about how we bring all the technologies together for patient benefit. I was in Norway, as I said earlier this week. They're working with other centres around the world on what is called in silico guided clinical trials, so modelling of the likely response in terms of clinical trials that could be more targeted. And interestingly, this brings together in Oslo groups in fields such as systems medicines, mathematical oncology and bioinformatics to really discuss the outcomes. And this is they're focused on breast cancer, and this is how they're bringing it together, but with a significant amount here of a design of multi-scale mathematical model of drug responses, through to comparing sort of what has been a mathematically modelled clinical trial to one that's sort of a standard of care type of clinical trial. But I put this in to say, in terms of personalisation, we need to think broad, we need to think about the engagement of other disciplines, and we need to think about how we use mathematical modelling and other approaches to areas such as clinical trials. I'm going to finish with the key challenges. In implementing a genomic future, we have to change end-to-end patient pathways, and we have to develop a workforce. And that includes an ongoing development of education and training that's fit for purpose, but that's flexible. We need to work within an ethical framework and really understand and have a debate with the public about public perception, but also public acceptability of elements of genomics. And you'll be aware of, for example, the recent media attention about the announcement that was made to whole genome sequence screen newborns. That's a real ethical debate that needs to be had in a much broader context. We have to retain and build public trust and create a contract with our people in our population to share and contribute their genomic data and ensure we've got the governance. And then we have to really think through how we reanalyse data, how we constantly go back when gene discovery is evolving at such a pace, and how we use polygenic risk scores and other evolving genomic technologies to properly bring to bear on our population. But in doing that, we understand the impact on services and an equitable offer. So this is my final slide, and I go back to a slide from Eric Topol to say where we need to head in terms of personalised medicine is this Google Earth map of an individual that brings together everything that we know that might be about social factors, about environmental factors, about the whole functional genomics pathway, but bringing that together with imaging, with, for example, radiotherapy data, and other diagnostic and clinical data, so that we really understand what personalisation means for each and every one of us. Thank you very much, and thank you for inviting me.